Hi everyone. Today we discuss the topic satellite DNA, selfish DNA and pseudogenes. Satellite DNA. Satellite DNA is the chromosomal DNA with a highly repetitive nucleotide sequence. It is found in highly heterochromatic and non-transcribing regions around the centromere of eukaryotic chromosomes. The monomer length of satellite DNA ranges from 150 to 400 base pairs. It was proposed by Peck et al. It is common occurrence in plants and animals but not in lower fungi such as yeast. The amount and sequence arrangement of satellite DNA varies significantly between different species. Satellite DNA can replicate but cannot normally transcribe. This is probably because its short sequence do not have promoters upon which RNA polymers initiate an RNA chain. Thus, satellite DNA is genetically inert and hence not indispensable. Satellite DNA contributes to essential process of formation of crucial chromosome structures, heterochromatic establishment, dosage compensation, reproductive isolation, genome stability and development. It is classified into mini satellite and a microsatellite. Microsatellite is the type of repetitive DNA in the eukaryotic genome with a 2 to 6 base pairs long repetitive sequence. They are widespread throughout the genome and can be used for paternity testing. Microsatellite is a type of repetitive DNA with 10 to 100 base pairs long repetitive sequences. Selfish DNA or junk DNA. This is randomly repeated and genetically neutral eukaryotic DNA sequence without obvious biological function. Though functionless, it can perpetuate, spread and accumulate in the species by virtue of its rapid replication and unequal crossing over. Curiously, it does not cause any survival disadvantage to the organisms containing it. Examples: spacer DNA and satellite DNA. Pseudogenes or false genes. Pseudogenes are non-functional segments of DNA that resemble functional genes. Pseudogenes have lost at least some functionality relative to the complete gene in cellular gene expression or protein coding ability. Accumulation of multiple mutations within a genome whose product is not required for the survival of the organism. Although not fully functional, pseudogenes may be functional, similar to other kinds of non-coding DNA which can perform regulatory function. The term pseudogene was coined in 1977 by Jack et al. Pseudogenes contain biological and evolutionary histories within their sequences. This is due to pseudogenes shared ancestry with a functional gene. Pseudogenes are a common feature of many eukaryotic gene families. The gene families of human actin genes, alpha and beta, globin genes, etc. Pseudogenes are of two general classes, namely orthophones and retropseudogenes. Orthophones, which are the dispersed solitary pseudogenes, which show almost all structural features of functional genes such as presence of promoter, introns, exons, and etc. They may be formed by addition or deletion mutation during gene duplication in multigene families. Example: globin gene family. Retropseudogenes are the defective DNA copies of mRNA. They have no introns, promoter and other sequence, hence they have no transcription and translational powers. Thank you.